Well, while the computer is being set up, let me just starting to thank for the invitation. It has been a pleasure to be here and at MIPT today and see the great development that is taking place. And I can see actually in different areas of research there are a lot of complementarities between what you're doing here and what we're doing in Germany. Okay, there it is. In Germany, we have begun a program 4 years ago, and I was asked to speak primarily about this, to be able to understand, prevent, and treat neurodegenerative diseases. This is a program which was launched by the German federal government, so is a federal initiative, and it is an institute, effectively, which is present at different sites in the whole country. The main site is in Bonn, and a number of other sites are present throughout the country. So let me tell you which problems we have in neurodegenerative diseases first and how we intend to address it. This is a slide that you see often when you hear about neurodegenerative diseases, and it simply illustrates that the disease begins much earlier than the symptoms are manifest. Often the disease begins 20 to 30 years before the symptoms appears, and this comes from studies in patients who are genetically predisposed. They are, for example, having mutations in the amyloid pathway, and therefore they tend to develop early Alzheimer's disease, juvenile Alzheimer's disease. And this, of course, tells us that we have to study not only the patients cohort, but also population cohorts and healthy mutation carriers. And to do this, also, we have to have a very strong cooperation between fundamental research and the clinical research, very strong interactions, because we don't know the causes of disease. In fact, probably the majority of dementias in general are not necessarily Alzheimer's disease, or do not necessarily present the alterations that we know in genetically transmitted Alzheimer's disease. So the problem in neurodegenerative diseases is that we don't have any disease-modifying therapy at the moment. We do not have very strong early biomarkers to diagnose the disease. The outcome measures in clinical trials are very weak, so we usually measure cognition, and it is extremely weak measurement. We should improve on this. And a number of large clinical trials based on existing hypotheses have failed, and particularly those clinical trials that have used cohorts of patients and they have tried to treat them with inhibitor of the amyloid processing pathway. And why is that? Why did they fail? Well, some people say because the theory perhaps does not reflect the disease. It may be the case that a number of these patients, or the stratification of these patients, is wrong, so it's a mixed form of dementia. The main reason that people ascribe to the failure of the trial is the fact that patients are treated at a late stage. It is too late when the treatment is initiated to be able to have a positive effect in a clinical trial. So there are some situations at the moment where obviously patients are treated in a preventive manner. There are some preventive trials which are ongoing, particularly in populations that are genetically predisposed. And those will tell us whether, of course, addressing the amyloid cascade may in fact have a positive or no effect whatsoever. So how do we solve the diseases, and why did the German government create a Center for Neurodegenerative Diseases? Strategy drives structure. This is something I'm sure you have heard before. And the idea here was to create a center which would make the best of the existing skills in Germany from the different places where neurodegenerative diseases is a focus of research, but then to add on top new expertise, to bring together scientists from different parts of the world. And to do this, actually, we had to create a structure where there is a critical mass of scientists involved in delivering translational research. This has to be coordinated within a single research organization for strategic reasons, of course, to be able to deliver strategy, to be able to implement a strategy which will produce translation. You have to create a structure where scientists identify themselves with. A simple network does not do that. You have to create a structure where the scientists who are part of it, who are already part of different organizations like universities, for example, identify themselves, which means creating quality. A structure which enhances cooperation and promotes high scientific standards and drive, of course, innovative research that can be reviewed very effectively for performance in science and administration, and that above all has a long-term support. You don't solve disease problem by having short-term grants. And so this has been an idea that the German government has taken on board and has decided to fund an institute which therefore is long-lived with a very strong strategic focus. So our funding is renewed every 5 years after peer review, but at the same time there is a substantial infrastructure which has been created throughout Germany to enable this long-term translation. So how are we organized? Well, we were founded on the 3rd of April 2009, and when the center was founded, actually I moved from the UK to Germany. We were like 3 people at the time. So myself, the administrative director, and the head of personnel, and of course a number of existing colleagues who had not been yet somewhat merged with the DZNE in Germany. And now we are about 700 people in the entire organization. We have, initially we had 7 sites, now we are 9. The funds come from the federal state and also from the federal government and from the state governments. And we do not give grants effectively. We are a single intramural organization that cooperates with universities and other public and private partners. Today we have, 4 years later, we have published over 1,000 papers, of which about 12.5% are in journals of impact factor above 10. So there is a substantial productivity which has been reviewed recently, very positive. But you know, science and translational research is not about publications only. Of course, publications are very important, but what we want to do, we want to be effective at delivering translation. And translation, often it is painted as a linear or a sort of a bidirectional line. The way we see translation is more like a cooperation between different research areas like healthcare, population, clinical and fundamental research. And our challenges, of course, are to determine disease etiology, to establish better models for disease, develop strategy for early intervention, better outcome measures and clinical trial design, and also better patient care strategies and healthcare policies. So the spectrum that we cover is quite large. The way we built this is to be able to create an infrastructure first. This is a common way to do it. So by doing a common infrastructure means taking the expertise which is available at the different sites that compose the DZNE and build on them. For example, we have now high throughput, high content facilities both in Bonn and in Tübingen. Here geared primarily to genomic analysis. We have antibodies and transgenic mouse and zebrafish facility in Munich, and so on in the entire country. And we work as an integrated organization, which means we have systems by which we can run the assays across the entire organizations. And at the same time, we have created first in Germany, which is a clinical structure which will enable national clinical trials. Of course, there are national clinical trials which have been carried on over years in Germany in a number of diseases. However, we have a major problem. We don't have registries in Germany. For cancer, there is one which has been created, and for other diseases are being created. But because of the way the healthcare system is organized, there are no national registries. So by creating a common platform, common SOPs, common ways of investigating patients with neurodegenerative diseases, we have now begun to create a registry for the major neurodegenerative diseases. And under the leadership of Thomas Gloggetter, who is our clinical director, we have now a number of national programs. You see, we have clinical ataxia, vascular dementia, and a number of other diseases. We are part also of DIAN, which is the dominantly inherited Alzheimer's network. And recently we have begun 2 studies, MIGAP and DELCODE, which will be the basis for a registry for Parkinson's disease and Alzheimer's disease in the country. And of course we are running, or we are planning to run, interventional studies. We're also very active on the international scene. I wanted to show you this slide to tell you a little bit about COEN. COEN is a Center of Excellence initiative which was initiated when I moved to the DZNE from the UK. It was initiated as a part of a collaboration with the UK, but then attracted also the Canadian Institute of Health Research and a number of other countries in Europe, and has given rise now to the countries are shown here. So we have at the moment Ireland, Germany, the UK, Belgium, Ireland Foundation, Italy, Spain, the Slovak Republic. So we have a number of countries that have joined this organization, and I think, you know, obviously If funding will be available from the Russian ministry, Russia will be very welcome to join. And the idea is that it makes it leverages centers of excellence. So in other words, you don't need to be able to invest substantial amount of money. You need to invest certain amount of money in one center or two to be able to enable collaborative projects. And this is now working extremely well. We are also part of the European joint program on neurodegenerative diseases, JPND, and we collaborate actively with American institutes like the Gladstone Institute and the Burnham that do research in this area as well. So let me just switch now and go a little bit towards the collaboration with industry. One thing is to collaborate with academic colleagues, and one thing is to collaborate with industry. Of course, academia has a number of characteristics. It's innovative and intuitive. It's driven by publication. However, it is accused of having poor reproducibility in their results. And of course, universities tend to have a relatively lack of strategy. This is normal because they have to cover a broad area. Industry, on the other side, has a systematic research approach aimed to applications, go or no-go decisions, and driven by pipelines and market strategy. Of course, industry is also adversely conditioned by a culture we live in, which is a risk-avert culture. I used to say that today, if you want to license aspirin, you would have a lot of problems because of the side effects. So obviously, there is this kind of risk-avert culture which prevents, in many cases, the introduction of new drugs on the market. So how do we foster an interaction between these two cultures? And there are different ways, and the way you are proposing here, I think, is a very promising one. We have basically identified two major areas. One is a pre-competitive type of collaboration, for example, to identify new output measures, new better outcome for clinical trials, where we can work together, where we can, for example, do a quick translation by using proof of concept type of studies. And the other thing, it is to be able to identify biomarkers, which are very important in neurodegenerative diseases, an area which is becoming more and more increasingly important. We have just heard about bioinformatics a few minutes ago, complex data management, and standardization of animal protocols and other protocols for clinical studies. And obviously, one second type of collaboration is the traditional one, to focus the collaboration on developing together new treatment and clinical studies. So which treatment for neurodegenerative disease is all very well. We can imagine good models, but where is the future? Where do we treat, or how do we treat neurodegenerative diseases? Well, I grouped here three different areas. In my view, I think it's a bit too early to drop the existing strategies which have been pursued by industry over the past 2 decades, which is trying to sort of interfere with the pathogenic mechanism of disease, like, for example, accumulation of misfolded proteins, A-beta, tau in Alzheimer's disease, alpha-synuclein or others in Parkinson's disease. I think we still need to pursue this for a certain period of time until we have a proof of principle that is either it works or it doesn't. However, this doesn't have to stop us to be able to address the major symptoms of neurodegenerative diseases, cognition, for example, which means synaptic plasticity, and finally, innate immunity or acquired immunity is becoming extremely strong as an area of research in terms of inflammation and immune responses. And I think we should probably get to invest a little bit more in this area in the future. And finally, I'll give you an example of what we're trying to do, which is hopefully the subject of a collaboration with MIPT in the near future to come. We're looking at the possibility to restore missing noradrenaline level in the locus coeruleus, or at least in the midbrain. So what happens usually in neurodegenerative diseases is that there is a very early loss of the locus coeruleus. The locus coeruleus is the sole source of forebrain noradrenaline, and it is a site of early tau deposition. Tau is one of the pathological proteins in Alzheimer's disease. It degenerates, as I said, very rapidly. So if you look in Alzheimer models and you produce a locus coeruleus lesion, you potentiate A-beta induced inflammation, you impair microglia, you impair synaptic function, you aggravate behavioral deficits. So of course it is an obvious sort of low-hanging fruit to try to restore noradrenaline level in brain and see whether we can get an amelioration of symptoms. And of course, this can be imaged very well. These are 70 images which were collected in Bonn. You can image very well the locus coeruleus. So you can select or stratify your patients based on the lesion that they have. And there is a drug which can restore noradrenaline level in the midbrain. This is called droxidopa or L-DOPS. And in experimental model system, it ameliorates behavioral deficits without touching the pathology of the Alzheimer mice, or at least of the transgenic mice which were used in this study, which modeled certain features of Alzheimer's disease. So there is a promising avenue to be able to use this drug in patients, as of course a drug that has gone through safety, so it's a typical exercise of drug repositioning and may give benefit in the short term. There is an evidence in human beings that this may be the case by using a different type of precursor for noradrenaline. In this case is the classical L-DOPA, which improves decision-making in adults with compromised cognitivity. So there is a possibility to restore this. This is also coming from one of our sites in Magdeburg. So future research, that's my last slide. So this is one example, but where are we going? Where is actually research going overall? Are we changing our way to make research? And that I think is strongly the feeling I have. We are at the sort of a borderline of a new period, a new conceptual way to do research. Obviously, direct extrapolation from animal and cell studies to humans, at least in complex diseases, is problematic. And many clinical trials based, especially in the brain, on the success in animal models have failed. And of course, that there could be complicated reasons for this, but definitely this is a matter of fact. And of course, I mean, we went for a long time with this idea, say, well, don't worry, I had the same thing and they cured me, the mouse tells to the little patient. But effectively, I mean, this has not happened. We cannot extrapolate directly, exclusively using animal studies. So there is one way of getting over this, getting over the deterministic way that biology and medicine have been actually done over the past century, I would say. And that is to be able to use pattern analysis. Pattern analysis integrates in a single database perturbation of different systems, which can be a fly, can be the worm, can be human beings, can be anything which basically we can analyze, and then using algorithms to try to find common nodal points which identify key areas that may actually be important in disease. And then this system can be interrogated, for example, in high-content screening facilities by using phenotypic assays with drugs or with biological microRNAs, for example, or so, to be able to identify pathways. It is amazing that in Alzheimer's disease there has been a recent paper published in Cell using this type of technology and predicting that the most important determinant in disease are factors of the immune system and inflammatory factors. And the very recent GWAS analysis study published in Nature Genetics last week came up exactly with the same answers. So I think we have to change a little bit the way we approach disease in terms of studies, especially for very complex diseases, different for monogenic diseases, but for things like Alzheimer's, Parkinson, diabetes, complex diseases that we see these days, we have probably to change the way we operate, and therefore cooperations and integration of different areas of research is essential in the future. And I'll close it here, and thank you very much.